So this is the concept review for limiting reagent lab. So let's say you have a reaction, it's a precipitation reaction. Let's say you have mixing solution A and solution B is going to give you a precipitate C and some other substance. So C is your precipitate. Okay. The goal of the lab is to figure out the concentration of each. Remember, you can't do that in a single experiment. If you want to figure out the concentration of A, you need to perform one experiment. If you want to figure out the concentration of B, you need to perform another experiment. So let's see the concept. Let's say you mixed equal volumes of A and B. So I'm going to draw two boxes equal in size to represent equal volumes. Now the solution A is going to have some moles of A. So I can't draw moles, so I'm just picking particles of A. So let's say 3 molecules of air is present in this volume, and B may be more concentrated. The two solutions may not be of equal concentration. So let's say there are some moles of B in this volume that you use to react, and let's say they're not equal. Okay, one could be higher than the other. Now think about how much precipitate you should generate when you mix those two volumes together. The ratio between A and B is 1 to 1. 1A reacts with 1B to produce 1C. So I'm going to do that visually. A and B react to produce C. And this is going to happen until one of them runs out. So A and B reacts to produce 1C according to the 1-to-1 ratio. And now all A is gone, so that will stop the production of C. Now, the question is, let's say you don't know how many A's are there in this volume, and let's say you don't know the moles of B in the volume of B that you used. How do you figure that out? Because concentration is basically, if you know the moles, you divide by the volume to get the concentration. So finding concentration boils down to being able to predict how many moles of that substance is there in that volume that you used. Well, let's see if we can backtrack it from C. So if 3 moles of C, or in this case 3 C's, is generated, so since the ratio between A and C is 1 to 1, so 3 C's are generated means 3 A's are used. Now let's go to tracking B. If 3 C's are produced, since the ratio between B and C is 1 to 1, 3 C's are produced would mean 3 B's are used. Remember, the stoichiometry will only tell you how much is used. Okay, it's not going to tell you how much was truly there. But the amount used for A is the amount that is there in the volume. Okay, because A is used completely, the amount used is all that there is, because all of A is used. So you're able to predict the moles of A from a back calculation only for the limiting reagent. For the excess reagent B, here B is the excess reagent because it's present in lots of higher quantity. So for B, for the excess reagent, you're not able to tell all the moles that are present in the volume. You're able to account only for some of them, the ones that have been used. So you cannot find the concentration of B from this experiment. You can find only the concentration of A. Okay, so if you want to find concentration of B, you have to do a separate experiment. Okay, that is basically the last question. It's asking you to figure out the concentration of the other reagent. And remember, you cannot use the previous experiment, the experiment you did in the beginning, to figure out the concentration of the other reagent. You would need to do another experiment. I hope this makes sense. Please fix your mistakes if you realize that you were not in the right track, and submit the lab in time.